to take a few minutes to talk a little bit more about membrane transport, and I'd like for you to take some notes. We're going to add these to our notes that we've taken in class or at home on the cell membrane and on osmosis and diffusion. So we've already discussed about membrane transport with the idea that we're looking at particles, water moving in and out of the cell membrane. There's a couple more things to add to that, and it refers back a little bit to some of the activities that we've done, or maybe you've seen online about the cell membrane, specifically looking at osmosis. So if we have a cell sitting in a solution in a particular cup, we will notice things that could happen to the cell. So for example, let's say we had 90% water inside the cell and we have 90% water outside of the cell. And so what's going to happen is we're going to notice water moving in both directions across the membrane. Some water is going to go into the cell, some water is going to leave the cell, and it's going to do so in a somewhat equal fashion. And as a result, there is no change in the cell shape or size. And the solution, therefore, is what we call an isotonic solution. That is talking about the solution that's sitting here in the beaker. Let's say we have another example of a cell, maybe sitting in a cup. And let's say we notice that the cell, the result of the cell sitting in the cup is that the cell shrinks, or it decreases in mass. The reason why this would be happening is because water would be leaving the cell. And the reason why water is leaving the cell is because there was a higher amount inside the cell and a lower amount of water outside the cell. If there's a lower amount outside the cell, that must mean that there's more room for a solute. And so the solution is called hypertonic. And then the third situation that we might actually see is in a particular container, we have a cell, and now let's say, as a result of sitting in that solution for a while, the cell gets bigger. Maybe we notice that its mass increases. Well, the reason why the mass would be increasing is because water is going into the cell. The cell. And if it's going to go into the cell, it's because there is a higher amount of water outside than inside the cell. And if we have a lot of water out, that means we do not have a lot of room for solute. And so as a result, our solution is hypotonic. Now you might be thinking, we've already talked about this in class. Well, the reason why I brought this up is because I wanted to talk about some special cases. We just mentioned generic cells here, but in reality, this can happen to both plant and animal cells, and it makes a big difference when it happens to a plant cell as compared to an animal cell. If you recall, one of the main differences between a plant and an animal cell is something called a cell wall. So if we have a plant cell, we have a cell wall that presents an outside barrier, very protective, pretty rigid, and right inside that cell wall, so close that sometimes underneath a microscope we can't even see it, we have something called the cell membrane. And I'm actually going to flip my arrow like that so you can see that. The reason why the cell membrane is so close to the cell wall is usually because somewhere in here we have a pretty large vacuole that's filled up with water, and it puts pressure on that cell membrane, and the pressure then forces it up against the cell wall. Now, in an animal cell, hopefully you guys are all thinking to yourself, well, in an animal cell, we don't have a cell wall. We simply have a cell membrane. And because we just have a cell membrane, we don't have this firm outside barrier to give us a square shape, or also to prevent that cell membrane from getting even bigger to the point that maybe it actually loses its capability and bursts. So I wanted to show you some pictures of what could happen in some of those cases. So let's say, for example, we have a red onion. I'm sure many of you have seen this before, eaten these before. Well, this is a plant cell, and I like to use the red onion because in class under a microscope we would actually be able to see the red coloring in the cells. It looks pretty cool. And it actually looks, in normal cases, like this. These would be typical skin cells from a red onion. We see a nice color. You can even see a nucleus there. We see this white boundary. Again, that is the cell membrane and the cell wall really close together. But if I were to put maybe some salt water on this solution and make this cell sit in salt water for a while, that is a hypertonic solution compared to the cell. And what I would notice maybe after about 5 minutes or so is that that red color suddenly, let me see if I can focus out a little bit better for you guys. I can't see the boundaries here. Let me bring it closer. Sorry about that. Just want you guys to see. Nope, it's not going to work. Let me kind of draw them in. The boundaries of the cell are still where they were before. But if you notice, the red color is no longer pushed up against the boundary like it was over here in this picture. What's happening, I can show you a cartoon picture of this, is that we put our cell, our plant cell, in a hypertonic solution, and it went through a process called plasmolysis. In plasmolysis, what happens is the water is leaving the vacuole because of osmosis. It's in this hypertonic solution, so water went from an area of high concentration to low concentration. And as a result, the cell wall did not move, but now we no longer have pressure from the vacuole because it's like a deflated water balloon, and the cell membrane has pulled away from the sides of the cell. The cell wall is still in place, but the cell membrane has shrunk because we don't have pressure inside. And so that's what we would notice in a plant cell. We still might see these rigid outside boundaries under the microscope, but inside we would notice a congregation of the materials, like what we saw up here in the red onion. If I can also show you one of an Elodea plant. We've talked about Elodea a little bit. This would be an example of an Elodea cell under a microscope. We see a nice even distribution of the green color. And then if I put some salt water on it, after a couple minutes, I'm starting to see that green color become concentrated. And after about 5 minutes, it becomes really concentrated. And just like this cartoon picture in here, we basically have a shrinkage of the cell membrane, a loss of pressure, and everything is congregating now in the middle. And so the evidence for that is what we see over here under the microscope. Now the cell membrane is here and not up against the cell wall, which is still out here. So hopefully that helps a little bit. Now, a plant cell, we could kind of reverse this process, and we could put a plant cell in a hypotonic solution. This would be an example then where we have more water outside and very little solute, and as a result, the water wants to go into the vacuole. The vacuole is going to love that. It's going to store that water for the plant cell, and the plant cell is going to get nice and big. If you notice, we have some bulging of the cell wall, but not a lot. The cell wall really doesn't want to move, and so as a result, the cell wall pretty much stays the same, but we also see a nice distribution of the color because the cell membrane has a lot of pressure. We call this turgid. So turgid is when the plant cell is in hypotonic solution. Plasmolysis, or plasmolized, is in a hypertonic. Please note that these are just talking about plant cells. So let's now compare that to an animal cell. So if you remember, an animal cell does not have that cell wall to try to keep things in line. And so I'm going to show you a picture of a comparison between the two. The top pictures are actually looking at a red blood cell. The bottom pictures are looking at a plant cell. So we just looked at all of these pictures. Well, we looked at turgid and what they call it shriveled, or plasmolysis, you can see written down there. But let's take a look at what could happen in an animal cell that is missing the cell wall. So we have a normal isotonic solution. Let's say we put it in a hypotonic solution. That's when water is going to be going into the cell. In this case, we now have the potential of bursting the cell wall because so much water is coming in, and I lack that cell wall to keep that membrane under control. We call this lysed. In this case, we are putting the animal cell in a hypertonic solution. So maybe like a lot of salt water or sugar water. And as a result, water is going to leave the cell, causing it to shrivel. So we have the word shriveled again, but they also have the word crenation. So what I wanted to do is add to our notes so that we can use some of this vocabulary word when we're talking about our membrane transport. So for a plant cell, we have the words turgid. So this is more pressure due to water moving into the plant cell. So turgid was more pressure because water is moving into the cell. That means it is in a hypotonic solution. In an animal cell, when it's in a hypotonic solution, we call that lysed. More pressure and possible burst of the cell membrane. And we called that lysed. It is in a hypotonic solution. If you remember, I used in class the idea that hypo gives you a hippo. It makes the cell get really big. Now, on the opposite side of that, we called it plasmolysis. When water leaves the cell, causing pressure to decrease. That would mean it is in a hypertonic solution. And then if you remember, for an animal cell, we called that crenation. Water leaves and cell shrivels. And again, it is in a hypertonic solution. Sorry about that mistake right there. So the vocabulary words I want you to remember are turgid and plasmolysis with plant cells, and this is because of that cell wall. And I want you to remember lysed and crenation with animal cells. Okay? You should be able to associate the word turgid with hypotonic, plasmolysis with hypertonic, lysed with hypotonic, crenation with hypertonic.